the DNA double helical structure. Normally, how many types based on particular one coil, one particular coil and all, or one complete curve and all, based on arrangement of nitrogen bases and all, it must be depends upon arrangement of nitrogen bases, whether it is one complete curve. Some of these DNA is present as 10, some other it is present in different types. It's present in the double helical structure. Before that one, what is the types it is present? Most common type, very important exam point of view. What is the most common type of DNA present most of the organisms? Then your answer is, they give options: aDNA, bDNA, cDNA, ddNA, eDNA, zDNA, like this. Many options they must give. Then you choose correct option. B type of DNA is the most common type. So for easy remembering purpose, I tell about in this one, that is zDNA as well as A, B, C, D and E. These are types, but most common type of all organisms, that is the bDNA. In this one, how many number of base pairs? How many number of base pairs present in one complete coil, one complete curve? Number of base pairs for one curve. That means here it is present, 12 are present. In A, 11 is present. B, 10 is present. It is 9. It is 8. It is 7 and all. According to this order, I arranged all these one for easy understanding purpose and all. But most commonly for one curve, it is present. That means one guide, G-Y-R-E. For one guide, it occupies 10 nitrogen bases and all. Then related to, in this zDNA, which direction this double helical strand one can moving and all, either right-handed position or left-handed direction of movement. Which direction? It is only left-handed direction. Left-handed direction. Remaining all are right-handed. Right-handed direction. All right-handed direction, as well as D also. Some of the cases, instead of right hand, sometimes maybe left hand also, left-handed direction also, it must be moving under related direction. Then for each and every guide, each and every curve, what is the diameter? What is the distance between each and every curve? The distance between that each curve. That means what is the distance? Each and every, the length of each and every guide and all. Or simply remember the length of each guide. What is the length? Here it is present. Normally in bDNA, the length will be present 34 angstroms. 34 angstroms are present. Related to zDNA, it is present 46 angstroms. Then aDNA, it is present 25 angstroms. Then cDNA is present 30 angstroms. Then related to C. Then DNA is present related to 24 angstroms. Like this variably present. Here there is no specific one because of, here I have written 7.5. Because of absence of one among the purine, Only two purines are there, adenine as well as guanine. Here, which purine is absent in E type of DNA? Normally, guanine is absent. Here, guanine is absent. Not only here, sometimes DNA also. So these are rarely present, D and E, because of may or may not be compulsory present in guanine and all. Remaining rest of it is present, but it is also rare related to Z. More commonly, that is A, it is most common. ABC is a common. Among this one, B is the most common type and all. What is the diameter? What is the diameter between the two helix and all? What is the diameter in that BDNA? The diameter 20 angstroms. Then related to here 23 angstroms. Here 18 angstroms. Here it is present 19 angstroms. Here they may not particular that why, because due to absence of that guanine sometimes may or may not present. That's why we can't say particular diameter and all. Specifically that D as well as E and all. That is related to their diameter and all. So this is very commonly is present, but exam point of view, for knowing about all this, I must inform and all here. But for exam point of view, B type of DNA. But which among these one that related to right-handed position? Which among these one left? Most of these one right-handed. Only one is the left-handed, that is the Z. But sometimes D also, apart from right, left-handed direction also, that it must be moving and down. Then between while formation of this DNA helix and all, then what is the particular distance present between nitrogen base as well as this one? While knowing DNA molecule, it requires nitrogen base, pentose sugar as well as phosphate. So what are the key role or which one act as a backbone while formation of this particular DNA molecule? Then you write as a phosphate. It acts as a backbone. Without this one, there is no formation of nucleotide. Why? Because nitrogen base plus pentose sugar, then it forms nucleoside. Nitrogen base, pentose sugar along with the phosphate, then it is called as nucleotide. That is important. But here, if it is eukaryote, that DNA molecule, it combined with histone proteins and all. That is octamer, H2A, H2B, H3, H4, such type of octamer. Normally DNA is the negative strand, negatively charged and all. Then it must be wrapped all along with positively charged of histone proteins. That is H2A, H2B, H2, that is H2A, then H2B. It is present like H3, H4. Twice it is present. These are four. Normally it is presented to the twice. So like this structure, like H2A. Why? Because it is normally it must observable in a tri-dimensional. Then one more H2A is present, like H2B. One more H2B, like H3. One more H3, H4, H4. Like this octamer. All along with that octamer, it is wrapped. The DNA molecule becomes into the wrapped and all. It is connected. There is H1 and all. This is histone octamer. This is H1 and all. Then histone along with the nucleus, along with the DNA strand, that is we call as nucleosome. Histone proteins along with the DNA strand we call as nucleosome. That is important. What is it by nucleotide? What is it by nucleoside? What is it by nucleosome? Very close related. Nucleosome. That is nucleoside. What is it by nucleoside? Nitrogen bases. Nitrogen base plus pentose sugar. That is nucleoside. Similarly, nitrogen base plus pentose sugar plus that phosphorus or phosphate molecule PO4 phosphate molecule, that is nucleotide nucleosome. What is the nucleosome? That DNA helix, DNA strands combined with histone proteins. DNA strand wrapped with, combined with histone proteins. It wrapped with, that is, suppose there is no histone proteins, that is the nakedly present. If it is present, it will be closed. Rapidly, it look like a beads on the string. All the nucleosome, if it observe on that microscopically, it look like beads on string. How small seed-like structure, like that manner, it must observable. Beads on the string and all. That is, then related to that particular structure, which one connected between nitrogen bases as well as sugar molecule. That is very easy under nitrogen bases. Normally DNA is present like this double helical structure. Like this is present. Here, one among this strand, it related to 3 prime, another one 5 prime. Then oppositely, whatever it may be present, here 5, then 3. Oppositely it will be comes under. Here, in these two particular, normally here discussing about, here we are discussing about related to B strand and all. Between two, these are nitrogen bases are present. Like this. To complete one from here to, after completion of this one up to here, this is one complete curve. This is one complete curve and all. From here to here, one complete curve here, that is 34 angstroms. Here between these two strands and all, nitrogen bases, between one nitrogen base and another nitrogen base, 3.4 angstrom. Between two nitrogen bases, it is present 3.4 angstrom. One complete guide. In this one regarding related to, it is present. One is the normal, another one is the deep groove. One is the shallow, another one is the deep groove is present. This is related to shallow groove. In this shallow groove, it is 12 angstroms. This is the deep groove. It is 22 angstrom. So 22 plus 12, that is the 34. So in that completion, one shallow as well as deep curve, deep groove, that represent, that indicates, that is one complete guide, one complete curve. Here the diameter, this is diameter towards this one, this is 20 angstroms between the two helix and all. But how it arranged in detailed manner and all. What type of arrangement? Normally it will be observing and all. Here, first you remember like pentose sugar. This is the pentose sugar. Here you labeling 4 and C and carbon atom. Then in this pentose sugar, there is a deoxyribose. The first carbon, it combined with here one among the nitrogen bases, either suppose adenine. Adenine, it is present normally, 2 are present. One is hexa and another one is the pentagonal. That's why suppose if it is adenine, then this adenine it connected with, it always connected with thymine with the help of double bond. It connected with thymine. So, one purine combined with another pyrimidine. That is according to Chargaff rule. It is also balancing and all. 1950 he said about. So, one purine combined with one pyrimidine with the help of two hydrogen bonds. Here, this is the first carbon. Here it will be connected with the ninth nitrogen. So, between pentose sugar and nitrogen base, this bond we call as glycosidic bond. N, why? Because N glycosidic bond. So between pentose sugar and nitrogen base we call as one bond is there, that is N glycosidic bond. One nitrogen base, another nitrogen base. How it is connected? With the help of hydrogen bonds. This is hydrogen bonds. Between adenine and thymine, two hydrogen bonds are there. Then this thymine, one more glycosidic bond, it connected with another pentose sugar. It connected with another pentose sugar. Here also first carbon, second, third and fourth. This is the fifth carbon. Fifth carbon and so here very important. Here between pentose sugar as well as one among these nitrogen bases and here that means pentose sugar combined with purine. What type of N-glycosidic bond? 1, 9. Here first carbon, here ninth, already just now I drawn that one, ninth nitrosyl 1. That's why here 1, 9. Glycosidic bond between pyrimidine and here and pentose sugar. What type of N-glycosidic bond? 1, 1. Here first carbon, there also first carbon, it is connected. 1, 1 glycosidic bond between pyrimidine and pentose sugar. Between purine and pentose sugar, 1, 9 glycosidic bond. Here it is present and all. So here third, it will be freely present. That's why it is a 3 prime. Here fifth one, it ends with the fifth one. That's why it is called as 5 prime. 3 prime and 5 prime. Similarly, it is going on. Like for example here, that one more pentose sugar, it is connected with suppose guanine. Suppose guanine is there. Here triple bond is there. Then it combined with the cytosine. Cytosine. Here fifth carbon, here third carbon. So fifth and third carbon, the phosphate group, it is connected. Phosphate group is connected. Here also, the third as well as here related to fifth one, it is connected. So this bond we call as 3, 3rd and 5th phosphodiester bond. In between this bond we call as phosphodiester bond. That is 3 and 5th. 3rd and 5th one. Here also the 5th and 3rd one. So overall 3rd and 5th one phosphodiester bond. Phosphates, how it connected with each other, phosphodiester bond. Similarly all. Here also one more related to here 1, 2. Like this it is moving on here. One more 5th carbon. It is connected phosphate group. Then it is connected with like this. As usual, here any one example, like instead of here thymine one can comes here, first one, then it is same. It is related to connected with adenine. This adenine, it is connected with, then as usual it will be presented like this manner. Similarly it will be present, but here you must remember very carefully, like this everything is present. What is the diameter? What is the distance? What is the distance between two that hydrogen bonds? In that hydrogen bonds, between hydrogen bonds of nitrogen bases, this is 3 angstrom. Then the pyrimidine it is present 3.3 angstrom. Actually, more than this it is present here. You must, for understanding purpose, I drawn elongatedly. This is little bit narrow. Actually, this is smaller, this is elongated one. Understand? But it is present 4.7 angstroms. If you calculate all these one, that is 11 angstroms. So, between nitrogen bases, what is the diameter? 11.1. Between the two helix, from here to here, this is 20 angstroms. Adjacent one, 3.4. That is very important. Between hydrogen bonds, similarly, that pyrimidine, wherever pyrimidine, here pyrimidine is come. Suppose one more area pyrimidine is come here 3.3, there 4.7. Wherever purine, because of two structures are present, one hexa, one pentagonal, that's why little bit elongated, little bit elaborated is present, purines and pyrimidines, only single, that hexagonal ring is present, little bit diameter will be the very less when you compare to the purines. That is 11 angstrom, that is total diameter in the B type of DNA, most of the organisms, there is a 20 angstroms and all. So like that, in that DNA structure, it is present, these particular diameters, the length, in that most of the cases, in that BDNA and all. So these are, in this along with the nucleo, just now I said, related to, it look like a beads or stringes and all. Beads or stringes. This is we call as nucleosome. Along with the DNA, along with the DNA helix, along with the histone octamer and all. Histone octamer is the positively charged. That DNA helix that related to the negatively charged, both are combined, it's present and all. This is that DNA link or linker DNA. Then between the two we called as chromatosomes. There is, so total, if you observe in that microscopically, like this it is present throughout. So each and every, any one among the area, if we expand it, so all these beads or stringes, like this it is present, each and every it is present, any one among this one, histone octamer along with helix we called as nucleosome. Between two nucleosome, linker DNA. Between the two, among these nucleosomes we called as chromatosomes. So like that you must remember carefully. In that first subtopic, the DNA, it is very important. What type of organisms? Double-stranded one. Along with this one, that is related to in RNA, it is most of the viruses on the genetic material. Non-genetic material, it is present like rRNA, mRNA, as well as tRNA. Apart from this one, the hnRNA, it is present. then small nuclear, then along with the major one, it is also present. It is also represent very clear importance and very important for transmission of that helpful but transmission tRNA. Then what is it by central dogma? Central dogma, there are crick is there as well as feminism is there. Central dogma in that sense, DNA, the replication, then it forms into the mRNA, then it forms into the proteins. This is central dogma. Who is the scientist expert about this central dogma? That is Crick and the Crick scientist. Similarly, DNA replication, then mRNA, then proteins. But here, one more. Here, transcriptase. This is transcriptase, this is reverse transcriptase. Here, mRNA, again one can formation of DNA. Some of the viruses like HIV, even after formation of RNA, why? Because RNA is a genetic material for HIV. Then once again it will be reversely formed into the DNA and all. This is the scientists Temin and Baltimore. Two scientists are exploring. That's why it is called as Teminism. This is we called as Teminism. So both Temin as well as Baltimore, two scientists are explaining related to this one. Then what is it by microsatellite DNA? What is it by related to mini satellite DNA? Microsatellite means 1 to 6 base pairs repeatedly present. 1 to 6 base pairs repeatedly present, that is we called as microsatellite DNA. Mini satellite DNA, 11 to 60 base pairs repeatedly present, that is mini satellite DNA and all. Micro as well as mini. So there is important, once again, microsatellite, micro. It will be present 1 to 6 base pairs, repeatedly present. Mini satellite, 11 to 60 base pairs, repeatedly present. Microsatellite and mini satellite and all. Some of the ciliated organisms like paramecium, 2 nucleus are present. One among these one act as a very important role for genetic one. That is micro. That is 2 nucleus is present, micronucleus, macronucleus. Macronucleus for vegetative one. It is not there is no important role for transmission of genetic material, but important role transmission of genetic material only for micronucleus. Whereas macronucleus to control the vegetative function, then which among the nucleus act as an important role for reproductive function? Then your answer is micronucleus. So in paramecium, ciliated organism, which nucleus act as a genetic material or genetic control, genetical control or genetic transmission from one to another? Then your answer is the micronucleus. It is present in double but micro. Most of the cases single nucleus is present, but in some of the bacteria the naked DNA is present. But related to one among these DNA regarding related to that bacteriophages. Bacteriophages, it may be chance to transmit one bacteria to another bacteria. There is also possible to transmission of this one. Then these particular aspects, very important role related to the first subtopic of molecular basis of inheritance. Okay, we will continue for next class.